Thank you very much, Alex, for the very kind introduction and leaving a few minutes for my talk also. Thank you. It's wonderful to be here, to be back in Vienna. It's 4 years that I was here in a symposium. And to commemorate also Max Birnstiel, who is so important for you, but also for science and molecular biology in more general terms. You know, he was a great science manager, and I think the campus here wouldn't be the way it is now. It's not just the IMP, it's also his influence on attracting other institutions. But he also was a great scientist. And as far as my knowledge goes, he was the first to really isolate a gene at the time when there was no cloning really in the current sense. And it was the ribosomal DNA repeats. And of course, ever since the question was having a gene enhanced, what is its structure, what is its function, how is it regulated? And actually, it's still the same questions we are asking. And that is the topic of my talk today. How are genes really regulated? We know that it's to a very large extent the first step in gene expression, transcription, which is highly regulated. Other steps are regulated, but this seems to be a very central point of regulation for the cell and for organisms. And we know that transcriptional regulation underlies not only the development of organisms, but also many processes in adult organisms. If you just think about the immune response, a very rapid transcriptional response triggered, by pathogens that enter the organism. So transcriptional control is something very essential, something very important, and it's really worth spending your scientific career, your life on this question: how are genes controlled at the transcriptional level? You may say, since this is such an old question, is there anything still to be done? And I want to tell a little story about Albert Einstein. I don't know if it's a legend or if it's really true, but the saying is that when Einstein was in Princeton giving lectures and teaching students, he also gave out questions for an exam. And then, of course, the academic year passes, and then the next exam, he gives out the same questions. And then one of the young people got him, Dr. Einstein, but you gave us the same questions for the exam as last year. And then Einstein said, don't worry, the answers have changed. And it's a little bit similar here. And I think one reason why the answers are changing is, of course, technology. When we, realized that a couple of years back, and we also had the funding to start with new technologies, we not only, extended our capabilities in structural biology to really do integrated structural biology and use different technologies, but we also went to in vivo top-down approaches using functional genomics, also developing the protocols further. And of course, the glue between the two, between the bottom-up and the top-down approach, is computational biology. So you can, for example, identify a mechanism using biochemistry and structural biology, and then you can make a mutation in a cell or deplete a factor and so forth, and you can perturb the system and read out in vivo genome-wide what the effect is. Or you can, collect data related to transcription, occupancy profiles, newly synthesized RNA on a genome-wide level, and then you correlate the data sets and you generate a hypothesis, and then you can test that hypothesis in the test tube. And we are doing this now for many projects, and it's actually very satisfactory, this approach, because essentially you can address all the questions you may have with respect to transcription and its regulation. Now, 10 years back, we produced the first movie of RNA polymerase II transcription, where you could visualize what is at the heart of the process of making a pre-messenger RNA. And just to remind you, this has been used in teaching now for many years, that there's a DNA template, obviously, that instructs the synthesis of the pre-messenger RNA. A nucleoside triphosphate is bound to the active site, and then there's a phosphodiester bond formed, and the mRNA chain is extended, and then there's a translocation step, and this whole nucleotide addition cycle is repeated, and this is basically at the heart of transcription. This is the chemistry behind transcription. And this was all done based on crystallography and biochemistry. And then, of course, over the last decade, there was, an explosion in structural biology due to the cryo-EM direct electron detectors, due to, better synchrotron sources, due to better proteomics, where we could do cross-linking, mass spectrometry mapping of the crosslinks. So we integrated the structural biology methods, and if I say we, then it's really the community, hundreds of teams around the world. And we applied it to the transcription machinery. And this actually allowed us to elucidate the different steps at the beginning of Pol II transcription, which are known to be the regulatory steps, the main regulatory steps in transcription. And this has been reviewed now, 13 years back by a great colleague, John Liss, where he summarized regulation of transcription at the beginning of genes in these different steps. Here you start with the opening of chromatin to make the promoter accessible. Then the pre-initiation complex can form at the promoter. The polymerase will be recruited to promoter DNA. Then the DNA can be opened. The polymerase actually begins de novo to make a pre-messenger RNA chain. It's then escaping from the promoter. And often it doesn't get very far. It's pausing about 50 nucleotides downstream of the start site. And then there's an exchange of factors and phosphorylation events. And the polymerase is then rendered into a complex that is highly processive, a complex that actually can go through chromatin very efficiently. And you have to imagine that in cells, in a normal transcription reaction, the polymerase in this state here, in the fully activated state, will go through about 10 to 15 nucleosomes each minute, and very few histones are lost in this process. It's an amazing question how that is possible. And I will actually end my talk with the first movie that is yet unpublished, which summarizes how far we understand that process of nucleosome passage. So we have, over the last decade, actually almost 2 decades now, tried to serve the community by filling in these different steps with mechanistic information. And that means, we solved structures of large, transient, multi-protein nucleic acid assemblies in different states so that we could see the movements and the exchange of factors and so forth. And we also did the corresponding biochemistry to a large extent to make sure that the structures we look at are actual functional intermediates. And that's basically the outline for the talk, but I will not go through all the steps. I will concentrate on a few rather recent and exciting findings along the way that are kind of exemplary for the kind of work that we are doing. And you will see that we arrived at some, I think, conceptual and fundamental insights that allow us to define much better the regulatory targets for thousands of transcriptional regulators. We actually know that the human genome encodes for 1,800 transcription-related factors, DNA binding factors that are expressed in different cell types, that can define the lineage, that can trigger the response to changes in the environment and so forth. And the idea we had, of course, was not to concentrate on these hundreds, even thousands of factors that have these specific functions and switch on particular genes or switch off particular genes. We really wanted to concentrate on the general transcription machinery, which is the target for these factors. Because if we understand all the intermediates in the reaction pathway, and if we understand also the energy barriers between them, I think that should be a solid foundation for later understanding how these many different activators and repressors of transcription actually function and what they do. So let's get started with chromatin opening. And we actually provided the first structure of a so-called pioneer factor bound to a nucleosome. This is a factor that can bind to nucleosomal DNA. And when it does so, this is SOX2, then it can also increase the accessibility of DNA to some extent. It actually uses its binding energy, that is the important point, to displace several turns of the DNA from the histone octamer surface and generate additional binding sites for transcription factors. And these pioneer factors are clearly distinct from traditional transcription factors, because most of these 1,800 factors will actually only bind to naked DNA. As soon as the DNA is wrapped up in nucleosomes, they will not have access. But these pioneer factors, maybe a few dozen or so, can actually bind to nucleosomes and begin to open chromatin. But actually, it's not enough to provide the binding energy. These factors also use very large intrinsically disordered regions to recruit other complexes. And what are these other complexes? Some of those are actually histone-modifying complexes, like acetyltransferase complexes, like the SAGA complex. It's a cofactor of transcription. Other complexes are chromatin remodelers that use the energy of hydrolyzing ATP to either shift nucleosomes away from the promoter or even to evict nucleosomes so that the promoter becomes fully accessible. And we also solved the structure of one of the first of these switch-SNF remodelers, which are doing exactly that in the promoter region. And from this, we can come up with a very simple model of how you expand the nucleosome-depleted region around the promoter. And this is that, a remodeler can either bind to the -1 nucleosome or the +1 nucleosome. These are the nucleosomes flanking the nucleosome-depleted region. And then these remodelers use hydrolysis of ATP to translocate the DNA and actually shift the nucleosomes further outside. If you inactivate them, then the nucleosome-depleted region will partially close again. This has been demonstrated by other laboratories. Now, the promoter region is now open, chromatin is open, the DNA is accessible, you can assemble a pre-initiation complex, but imagine you have to bring a lot of factors there, actually around 70 factors just to make the pre-initiation complex, and then many more if you want to start transcription and move into elongation. So how do you bring rapidly in a transcription response all these factors to active promoters? One possibility is, again, to use these intrinsically disordered regions and to form condensates at highly activated genes. And we found in several studies that the C-terminal repeat domain, which is actually drawn here to scale, it's about 10 times the diameter of the polymerase core, that the C-terminal repeat domain is very important in the recruitment of polymerase and also the activation of genes. If you make human cells which lack half of the CTD, they're still viable, but if you trigger a transcription response, it will take much longer to switch genes on. So there's a rate-limiting step of delivering all the components and especially the polymerase to active promoters. Now imagine you have all the factors there, then you assemble the pre-initiation complex. And that is really a project that we started in 2004, so it's almost 2 decades, and now we finally arrived at the atomic structure of the pre-initiation complex. This is the classical assembly pathway where you see the TADA box binding protein in red and binding of TF2A and TF2B that flank the TADA box. And this assembly can recruit the polymerase, which is in silver here, together with TF2F in violet. And then the promoter is retained at the polymerase surface by TF2E and TF2F, which form this kind of handshake above the DNA. So they encircle the DNA. Now the promoter is captured on the surface of pol 2. And a very large factor, TF2H, actually a 10 subunit assembly, is then joining on the downstream DNA. And this is the factor that has multiple functions, but in particular is important for DNA opening for transcription. And up here you have the mediator complex. That is a cofactor of transcription, but also generally needed for transcription initiation. It, for example, stimulates this complex here in orange, which is the CDK7 kinase. And what is so exciting about this is that, once you assemble the whole complex and the reaction starts, there's an inbuilt trigger to already disassemble the PIC. Because once you activate that kinase, you destabilize the PIC again. And that makes it very short-lived. It can form within seconds, but it also disassembles within seconds. But you have to be able to do that, right? That's an inherent issue in molecular biology. Once you have a functional assembly, you cannot freeze it, right? Otherwise the gene would be poisoned. You have to immediately have a second mechanism to resolve it again. And so this is what is done by mediator, by activating this kinase. But before it's doing this, there's a time window to open the DNA and start RNA synthesis. And we know now the atomic mechanism of DNA opening. I will show you that mechanism. But before, I want to emphasize that the pre-initiation complex actually is highly conserved from yeast to human. So we have now atomic structures both for the yeast assembly as well as the human. And for the human assembly, actually a number of laboratories have contributed, in particular Eva Nogales and Yuan He, who actually came from Nogales, and more recently the Shu lab in China also. So we have these structures, they're very similar, but it's still extremely useful to have both systems. And I show you two examples why it is so useful. First of all, technically it's useful to have both systems because sometimes you may trap a certain intermediate only for the yeast complex or only for the human. And this happened actually for the promoter opening intermediates. I will show you those. But sometimes it's useful to have both systems also because the biology is different. And I show you an unpublished example for that, and that is how the pre-initiation complex relates to the +1 nucleosome. So for the first question, how does the DNA open? We could use the yeast system to actually trap a very early intermediate where we see the beginning of the opening of the bubble. And this was just published. So TF2H has this translocase, this ATPase, which undergoes a power stroke to open the DNA. And we see the opening of the first few DNA bases in this region. You just, get rid of the Watson-Crick base pairing by taking the two strands a little bit apart. This is then stabilized by two loops here for TF2F and the polymerase. When you mutate those loops, then transcription is down by 50%. So it's extremely important to stabilize this early intermediate. And then, once you have stabilized that, you can extend the bubble in the upstream direction. Now, but what we couldn't do in the yeast system is to trap that translocase, the TF2H translocase that actually pumps the DNA towards the active site of the polymerase. But this we managed to do in the human system. We still don't know why, it's for technical reasons. But since the mechanism is conserved, the combination of the two gives us the atomic mechanism of DNA opening. And this is now just one subunit of TF2H, this translocase XPV, in different states. When it first binds ATP, it will induce a twisting motion to the DNA, and then hydrolysis is actually resetting the ATPase motor, and the twist is then propagated downstream, leading first to this intermediate, and then the opening continues in the upstream direction. Now, is that relevant at all in cells? The answer is yes. Just to give you an example, again in the yeast system, because it's simple, you have only 6,000 genes, only a few thousand are active. We use RNA labeling, and then we can get the RNA synthesis rates for all the yeast genes that are active. And what we find is that when we rapidly deplete the translocase from yeast cells using the anchor-way system, then 80% of the genes are automatically downregulated very quickly. But about 15 to 20% of the genes are still transcribed normally. And when you then analyze that sequence, where the DNA opening starts, you find that this is a sequence that is unstable and the duplex stability is actually low. So, some promoters evolve to be opened independent of TF2H. And this is another level of regulation that you see here. Now, the second example why it's important to look at the yeast and human system is differences in the biology. So you all know that there's a flanking nucleosome next to the promoter, the so-called +1 nucleosome. Here would be the nucleosome depleted region where the pre-initiation complex assembles. And there's a very strange observation. This comes now from functional genomics, from occupancy profiling in cells. And that is that the TADA box actually is at very different distances in different promoters from the transcription start site. It can be 40 base pairs upstream, can be 120 upstream. And the start site, however, is at a fixed distance from the +1 nucleosome. This is kind of counterintuitive because you would think, the pre-initiation complex knows where to go and then it starts at a certain point. But in yeast, it's different. It's like the +1 nucleosome knows where to go. It sits at a certain site and it defines where the transcription starts. And actually from here to the start site, there's a so-called scanning of the DNA. Very particular thing that you don't have in human cells. Now, we actually thought, well, then there's probably a possibility to get a structure of the pre-initiation complex with the nucleosome. And we managed to do that. It's still unpublished. It's the first structure of a pre-initiation complex in the chromatin context with a bound +1 nucleosome. You see it's in a defined orientation, and you see it's binding TF2H. There's 4 interaction sites. These are actually relatively loose loops, so this is not a specific binding of the nucleosome. It's more like docking to the nucleosome in a particular orientation, and we think that can explain why then the transcription start site is always at a fixed distance of the +1 nucleosome and not so much with respect to the TADA box, because the polymerase is scanning downstream for the start site, and apparently then when it hits the +1 nucleosome, there could be a barrier, and then it actually starts there. So this is the situation in yeast. But now if you look in human, again using data from different laboratories, Liz, Edelman, and others, what is the situation at human promoters? There you have a fixed distance from the TADA box to the start site. So the pre-initiation complex sits at a certain site and it initiates at a certain site, always the same distance from TADA. And the +1 nucleosome is further downstream. It's actually 140 base pairs from the start site. And the reason is that when the polymerase is released into the gene, it pauses at around 50 base pairs downstream. It pauses at this site, and that will be just in front of the +1 nucleosome. We don't think it pauses because of the nucleosome. We think there's a pause site, and the +1 nucleosome is found adjacently to the polymerase that is paused here. Now, can we get a structure of this entire thing? The answer is probably. We don't have it yet, but since we have structures of the PIC, structures of the pause complex with these pausing factors, and structures of the polymerase in complex with the nucleosome, we can actually make a model. And this is how this model looks like. It's very interesting because you take the in vivo distances for these complexes, and you can make a model very simply without any clashes. So this can really, it's a situation that can really exist. But the component that doesn't come in here is, of course, the kinetics, and I will talk about that later. It turns out the PIC exists only for seconds, whereas the pause complex can live for minutes or even longer. And that actually will tell you a lot about the regulation of genes. I will come back to that point. Now, we initiate, we go to the pause state. There's this negative elongation factor here in red, which stabilizes the pause state. We actually know how it's stabilizing it. There's a rearrangement of the nucleic acids in the active site. It's called the tilted stage of the hybrid. These are details, but we have analyzed that in great detail. But recently it was found then that this paused polymerase can be subject to early termination. So basically what happens is you initiate the polymerase pauses, but then it can be targeted by a complex called the integrator complex, 16 subunits, very large complex, and that can lead to termination. So the polymerase is released from DNA, can be recycled and initiate again. We recently were able to get the structure of the integrator bound to the pause state of the polymerase, which you see here. There's some induced fit. So for example, this subunit here gets folded and docks then to the polymerase. And the whole assembly is very interesting. It explains quite a few things. The first thing that it explains is how the integrator can recognize the pause state, because that was enigmatic, right? The pausing happens inside of the polymerase at the active site. How would you recognize whether that polymerase is in a pause state? Well, the way integrator does it, it actually binds to the polymerase, but also to the two factors, DSIF and NELF, which are characteristic of the pause state. And by making these multiple contacts, it can basically detect, this is a paused polymerase, this is the target for termination. And the second thing that is very interesting is that the integrator has one subunit. It's called int11, which is an endonuclease, and that subunit opens up when the integrator binds to the paused polymerase. And then an active site is formed, the endonuclease active site, that can accommodate the nascent pre-messenger RNA here in red. And actually it's 20 nucleotides from the 3' end of the RNA where RNA synthesis takes place in the polymerase active site, all the way to the endonuclease active site where the cleavage of nascent RNA takes place. Why is that leading to termination? Well, one issue is that, of course, when you cleave the RNA here, you lose the 5' cap structure, which is always found on pre-messenger RNAs and which is formed immediately after transcription starts to protect the pre-messenger RNA from 5' degradation. But now when you cleave here, you lose a few nucleotides that end the cap, and so you have a vulnerable 5' end. And that is probably one reason why transcription terminates, but we don't understand the detailed mechanism of how actually then the polymerase is opened up and how nucleic acids are released, but we probably get there because we work on such termination intermediate structures. It's actually one of the big unresolved issues in transcription in the field. Now, we understand, how you convert a pre-initiation complex to the pause state, and we also understand better how you can recognize that state and get to a termination intermediate. And the question is, of course, when you want to switch the gene on, how do you convert this pause complex into an active elongation complex? And this is done by the positive transcription elongation factor B, which is a complex that contains the CDK9 kinase. So CDK7 is a kinase that releases the polymerase from the promoter for the promoter escape. That is important. And CDK9 is a kinase that releases the pause complex and forms an actively transcribing elongation complex that actually carries also the positive elongation factor. We call this complex EC star, and it carries, the PAF complex, SPT6, and DSIF, which is this green factor here, and probably other positive elongation factors. So this is the complex that can now go through nucleosomes. And before I tell you more about this, I want to summarize, what we learn about the regulation of transcription at these early steps. And that is actually the take-home messages are simple, but conceptually it's very, revealing and simple and beautiful. And so the take-home message is the following. You start transcription, you go to the pause state, and then there's a decision whether there's termination, then you have to start again, or whether you release that polymerase into the gene body and then it passes through nucleosomes quickly and completes pre-mRNA synthesis. And the CDK9 kinase is the key to understanding this early regulation. And now what we did is the experiment, we used the Shokart cell line. There's a mutation in the CDK9 active site which leaves CDK9 intact, it's functional, but you can now use a small molecule, an inhibitor, and you give that to cells in a chemical biology kind of approach, and then you inhibit only CDK9, right, and no other kinase. And then you observe how the rates of transcription change, and we developed a multi-omics protocol to do so, and we can actually not only monitor the productive initiation frequency, which is like how often do you initiate and complete a messenger RNA. We also can then extract the pause duration, which tells us how many minutes on average the polymerase stays at this site. And this is possible by combining different functional genomics methods, occupancy profiling, RNA labeling, and using a mathematical model to fit those multi-omics datasets. And the next movie summarizes what we found. So now we inhibit CDK9, and what you would expect is, of course, that the pause duration goes up, right? Because you don't form this active complex anymore. And that is what we see. The pause duration is going up. Each dot here is for one active human gene. Pause duration is going up, but what is even more striking is that the productive initiation frequency is going down. And this was, it's a typical kind of thing where you do an experiment to confirm a hypothesis. You confirm it, but then you extend it because you also observe something new. So the initiation frequency goes down. And what does that mean? Well, that's the beauty that is summarized here. It sounds trivial in retrospect, but for decades this was debated how this could work. And the take-home message is, if the pre-initiation complex can form every few seconds, right, you could in principle initiate every few seconds and make a lot of messenger RNA over time. So you switch the genome. But since there's a pause polymerase that lives for minutes, it puts a limit to the initiation frequency. And if you want to have a higher initiation frequency, if you want to switch the gene on more, you have to decrease the pause duration. You have to deliver CDK9 to convert more of the pause polymerase into the actively elongating form. And then you can allow for higher rates of initiation because this limit is lowered, right? And this is summarized here. So the pause polymerase is an elongation complex, but this elongation complex can actually provide a feedback to initiation. And this is how you can explain that you can, by elongation control initiation and thereby control how much messenger RNA you make per time, how many mRNA copies you make per time. You can control the activity of the gene. Because, you know, in human genes, on average, it takes 40 minutes to transcribe them. So these genes wouldn't care whether the pause polymerase is there for 3 minutes or 5 minutes. Doesn't make a difference. But since, you know, it makes a difference on how often you initiate, depending on how long the polymerase is in the pause state, you actually it makes a huge difference in the number of RNA molecules you produce and a huge difference in the activity of the gene. It sounds trivial now in retrospect, but you know, it's only over the last 3-4 years that this has been realized. Now we're in a situation where the polymerase that is in front of the +1 nucleosome has exchanged all the factors, right? There was the phosphorylation events, the negative factor was released, the positive factors are now there. Now you have this EC star complex, but it's still in front of the nucleosome. And now you have to understand how you go through the nucleosome and through subsequent nucleosomes. And that is what we studied also over the last years. And basically we went back, you know, to the in vivo protocols. And what we did is we now very rapidly depleted these positive elongation factors from human cells. And then we observe, using our multi-omics approach, what happens to gene activity, what happens to the occupancy of engaged polymerase, what happens to RNA synthesis. And what we found, for example, just one example, when you deplete SPT6, one of these positive factors, and you look at the site where the +1 nucleosome is found, then you see that the occupancy with engaged polymerase is going up quite dramatically. So basically, when you take SPT6 out, the elongation complex has problems to go even through the first nucleosome. It gets stuck to a large extent in the first nucleosome. And you know, to analyze that in more detail, we went then back to the biochemistry and we derived a system that would allow us to study in a very defined manner which factors do you actually need to go through a nucleosome in vitro. So we can put the polymerase at the end of this nucleic acid scaffold and we can add nucleoside triphosphase and the polymerase will transcribe in this direction and it will transcribe into the nucleosome, right? And it will get stuck because the nucleosome is a barrier. And then we can add factors and we can find out which factors are really needed to make the polymerase go through the nucleosome. And basically what we found is DSIF or SPT45, TF2S, CHD1, and FACT, they all help. And when you have them all together, you get actually a lot of full-length RNA product, which means the polymerase went through the nucleosome. You know, if you don't have them, you can only get a little bit. It's probably a little leaky, the system, but hardly any product. Now, many of those were described already by the community as being factors that help the polymerase to go through nucleosomes. And how we can deal more with the mechanism and can go back and do structural analysis. And the first one I want to discuss is TF2S, and then I go briefly to CHD1 and FACT. And when we look at TF2S, I have to say that we generated this movie from a large number of unpublished structures of intermediates of the polymerase passing through nucleosomes. So there's still some science fiction here because we have to interpolate between the different structural states. But you know, it will be close to the truth because we have trapped several states. So the polymerase is a strong motor. It begins to unravel the DNA from the histone octamer. It can go in for several turns. And at some stage, H2A, H2B is lost because it doesn't have any DNA contacts anymore. So you generate a hexasome, right? The polymerase can go further even. But obviously, it's competing forces. You know, the histone octamer is highly charged. It wants to reform the nucleosome. So sometimes the nucleosome actually wins, and the polymerase is then going backwards. This you also see in the movie. And we've even trapped such a backtracked intermediate in a structure. How is that possible? Now you reform the histone DNA contacts downstream, but that means that the RNA that you just synthesized has to go through a pore beneath the active site, and TF2S now has to cleave that RNA fragment to restart the polymerase. And this beautifully explains why TF2S is needed, because it gives the polymerase the possibility when it gets stuck to try again, so that it can try again to go through the nucleosome. So that is how it's, TF2S works in nucleosome transcription. But what about CHD1 and FACT? Well, CHD1 is exciting because it's a single subunit ATP-dependent translocase. It's also a chromatin remodeler. And what we found trapping this intermediate structure is that it can actually pump DNA into the active center cleft of transcription. So it's like, a little motor that helps the polymerase to progress further when it gets stuck in the nucleosome using ATP. And what is interesting about this structure are actually two things. One is that you only get this defined binding when you generate an intermediate of nucleosome transcription. So we let the polymerase run into the nucleosome until it gets stuck. And then we add the remodeler, and then we solve the structure. And you see that some of the DNA is already unraveled here. And the second thing that's interesting is that an inhibitory domain in CHD1, which normally would bind the DNA, is released in that intermediate. So the model is the following. You can decorate a nucleosome with the remodeler. Nothing happens, right? Because there's this inhibitory domain bound to the DNA. Now the polymerase approaches, and it begins to unravel the DNA. It sets free this repressive domain and thereby switches on the remodeler in just that moment when it needs it to go through. And this is a model, but it's again such a simple model that probably it will turn out to be true. What about FACT? Well, Danny Reinberg has characterized that beautifully. It's a chaperone. It helps you to keep the histones around the site of transcription because, the polymerase can go through when it has all its friends, but then the histones may be lost, so you need these chaperones to keep the histones at the site of active transcription. And Caroline Luger has recently shown that FACT can bind to partially unraveled nucleosomes. And she solved that structure of the partially unraveled with FACT. And this structure that we have is quite similar. It's only shifted by one term, but we see very similar contacts of FACT to the histones. And you see this contact here, for example, is to H2A, H2B. And I think that explains why, in reality, H2A, H2B is not lost. I showed you the loss, but in that structure, we didn't have FACT, so we lost the H2A, H2B. And here, it's retained by FACT. A last thing, if the obstacle is really dramatic, so for example, if you have a UV lesion, DNA damage or so, then the whole complex unravels. I don't have time to talk about it. A new complex forms that you see here, and that has a translocase called CSB, the contain syndrome B, and that has this pulling pin. So it uses, again, ATP to actually pull on the upstream DNA. And when you pull on the upstream DNA, you also push the polymerase forward. So you see there's all these mechanisms to help the polymerase go through. You can pull on the upstream DNA to push it forward. You can put a remodeler in front that pumps the DNA in. And you have TF2S, which restarts the polymerase when it gets arrested due to backtracking. So it's maybe all these factors are needed to make sure that under normal conditions, these long human genes are transcribed to the very end. So far, I only talked about the synthesis of pre-messenger RNA, but you all know that pre-messenger RNAs are co-transcriptionally capped, spliced, cleaved, and polyadenylated. And I think this is all highly integrated. There will be intimate interactions between these different nuclear machineries. And with this in mind, we tried, in a collaboration with Reinhard Lührmann at our institute, to get a first structure of a transcription complex bound by part of the spliceosome. And we started, obviously, with the very first component that joins, which is the U1s nerve. So you don't need to know anything about splicing here. The only thing you need to remember, you start the splicing cycle with U1 SNP, and that is a very interesting one because there's high levels in cells. And we indeed managed to get a complex of the U1 SNP here on the left on the transcribing RNA polymerase II. And what is so exciting about this complex, not just that it's the first, where you have a transcription complex and a splicing complex in one structure. The most exciting here is that the mode of interaction is very unexpected. Everybody knew that the nascent pre-mRNA has, this kind of 5' splice site sequences which are recognized by the U1 as an RNA. This is, known for many, many years. But what was not known and very exciting is that there's a direct protein-protein contact between RNA polymerase II and the U1 SNP. And this led us to propose a speculative, but I think also conceptually very interesting new model about co-transcriptional assembly of a spliceosome. And that is shown here. We call it the growing intron loop model. And why do we do so? Well, simply because we think that through this direct protein-protein interaction, you can retain the 5' splice site of the pre-mRNA on the surface of the transcribing polymerase as the polymerase goes through the intron. And why is that important? Because it solves this long-standing mystery of how the two splice sites, the 5' and the 3' splice sites, can be brought into neighborhood, into proximity. Also on the sequence of the RNA, they're often 10,000 nucleotides away, sometimes more. These introns are huge. And now this model explains how, in a very densely packed nucleus, the two sites will find each other. Because by this direct protein interaction between the U1 SNP and the polymerase, you retain the 5' splice site near the exit site for the nascent RNA, and you form a longer and longer loop, very long loop can be, as I said, 10,000 nucleotides. But as soon as the 3' splice site then appears on the surface, it will be just next to the 5' splice site. Now it's a model, but it makes you think very differently of how the splicing process is initiated co-transcriptionally and how the two sites actually find each other. Now we went back in vivo, you can use pladenolide B, which actually stops the process here at the B complex, and then you find something very exciting. Talking about feedback, you see again that the pause duration goes dramatically up. This is again our multi-omics, and the initiation frequency goes down. So when you stop spliceosome assembly at this level of the B complex, the pre-spliceosome, transcription is downregulated. And you can then look, why is it downregulated? You find that there's more polymerase at the pause site, but less PTFB. So, the pause polymerase is stabilized because somehow the pause polymerase at the promoter knows that downstream something went wrong and, spliceosome assembly wasn't completed. And so basically, this is very interesting model where on a linear model, the polymerase the intron loop will grow and grow and grow, and you get then stuck when the U1 joins for forming this B complex, and somehow that is a signal to pause the polymerase in the beginning so that there's no new initiation. And we have no idea how, but one thing I can tell you is that most likely this is entirely wrong that they're all separated. Most likely it all happens in one super complex structure, and the DNA and RNA are going in and out, but these complexes all talk to each other, and there's yet another checkpoint. If you want to switch genes on or off, you have to switch initiation on or off. And as cells became more complex, new and new layers of regulation were added, which you don't have, for example, in these cells. And that is that elongation control in a backwards manner can also be used to control initiation and to control whether a gene is active or inactive. This is a traditional activation, right, where activators lead to opening of chromatin, lead to recruitment of factors, lead to pre-initiation complex assembly. This, of course, is very, very important. But now you have this additional layer of regulation. Again, a speculation, why should you do that, right? I think the reason is, if you think about the information must be stored somewhere. You have 20,000, 22,000 genes. Where do you store all that information for regulation? You cannot just store that in this short, region of the core promoter. Yeah, there you can dictate, does TBP bind strongly? Does the DNA open easily? That's fine. But if you can also evolve downstream elements, the pause site for the polymerase, the 5' splice site, even 3' splice site, and they all feed back to initiation, you have a universe of regulatory possibility. With this, I want to close. I want to just give you a tease of what is coming, because in the end, we would like to understand those silent genes, which are probably packaged in heterochromatin, but also active genes, ideally in their natural environment with all components. And this will not be possible by cutting through a nucleus and doing tomography. We tried, others tried. It's basically all black, right? It's totally packed. So how do we want to do it? Well, our idea is that we isolate chromosomes. This is, for example, a part of a polytene chromosome. And then with the new EM methods, with the better detectors, better software, and so forth, maybe we can do something that wasn't possible in the 70s, where actually beautiful images have already been obtained. And actually, those dots here are most likely nucleosomes. They have the right diameter and they have the right distance from each other. And so our idea is, and that was actually published a few days back, that we get some kind of chromatin secondary structure. So we solve single, particle structures of trisomes or tetrasomes, and then we try to fit those structures into tomograms of these higher order chromosomes, and hopefully we can get an idea of the topology of genes. And this is now the dream. Maybe we find also some of our other friends of these many large complexes in there when they're left over. Once you have explained all chromatin, maybe there's some densities left. So that's the next decade, or maybe 2. And I wish to thank all the people involved. Of course, I cannot mention everyone, but some key people, Sandra, Shintaro, Vasan, Clemens, Kayo, they were all instrumental over the years to get the pre-initiation complex project to what we have now, the atomic final structure. And then Carrie, she's also here in Vienna. Seychelles, Lucas, Saskia, Björn, they have been really instrumental for understanding the elongation control. Carrie started because she was the first to move our yeast projects to the human system. And then Seychelles got the pause complex and the release complex. And then Lucas got the first structures with nucleosomes and also got all these other projects started that actually led to the movie now together with Moritz Ochmann and others. And Saskia and Björn developed the multi-omics approach. She's experimentalist and he's a bioinformatician. He did the mathematical modeling. Then Isaac is the hero who made the integrator complex and then got also the structure with integrator. And Su Yang is the one who got this U1 structure. She will take the whole project and she will be the co-transcriptional splicing person taking a group leadership at LMB actually in August. Christina is a great functional genomics person, as well as Livia. They were all working on elongation control. Sarah is the bioinformatician and Michael also long-term computational biologist. Christian is the scientist who keeps the EM at the highest level and is developing the protocols. And Haibo is an amazing postdoc who got this first structure of a nucleosome on the pre-initiation complex, but before already got interesting other structures. And Henning is our long-term collaborator who is doing the mass spec for the cross-linking, which is important for the integrated structural biology approach. And maybe if there's some students who now think, well, that's a huge amount of work and so forth, it's true, because if I look at the past, people who went through the lab and they all basically contributed to get us to the stage where we are now, crystallizing individual mediator subunits, then 2 subunits, then next PhD thesis 3 subunits. So, don't worry. I mean, we all give our contributions, and then over the decades, there's a more complete picture. But there's so many things out there to be discovered. It's just wonderful. Science is an endless frontier. And with this, thank you so much for having me, and thanks for your attention.